Now we're ready to go through section A, which is counting the red blood cells. You can see we have the different pieces of equipment that we need for this lab right here, as well as the instructions. Just to go over what we're looking at here, we have our pipette tips for the automatic pipetter, cover slips, the automatic pipetter set to 10 microliters, a hemocytometer with a cover slip on top, and we have some pre-diluted blood. So this blood's already been diluted for you, and also a counting device. So our first step is to shake the microtube with this blood suspension well to resuspend the cells. So these cells are quite heavy and they tend to settle down at the bottom of the tube. So it's a good idea to shake that up just to resuspend them to make sure you're getting an accurate count. Now we're going to take a 10 microliter aliquot of that suspension using our automatic pipetter. So again, we have it set to 10 microliters. I have a pipette tip set on the top there. And here we go. Just pushing this knob down and pulling up to get my 10 microliters. Next, we will load up the hemocytometer with this blood. Okay. So you can see the blood has moved across the entire side of the hemocytometer. So each side of this hemocytometer has a little grid on it. Right? And so we want each side we want to be able to do a count on. Okay, so I'm also going to load up the other side so that we have some extra blood ready for the count. There we go. Okay, so again, it's quite important to make sure it spreads across the entire grid. There. Now this pipette tip I'm going to put into biohazard container. Okay. We're going to let this settle for a few minutes and then take a look. In the meantime, we're going to go through some instructions on how to use the hemocytometer. So right here we have our hemocytometer loaded up with blood, and that's just sitting there to allow the blood to settle. Here's a diagram to represent that same hemocytometer. And what this shows is the grids that you would actually see on this hemocytometer if you zoom in on it. So the hemocytometers contain a series of grids within grids, so some sort of nested grids. And the reason for that is to provide different types of grids for different types of counting, depending on what you're looking at. So both the size of the cells you're looking at and the amount or quantity might influence which grid you choose. In this case, because we have a lot of red blood cells and they're quite small, we're going to be using these little grids in the center. We're going to be counting all of the red blood cells in 5 of these little grids and then putting that number down in our table. If you look over here, you can see a closer view of one of these little grids. Here are some rules for counting. These rules are in place to ensure you count the correct volume of cells. If you were to count all of the cells touching the edge of each little grid, you would be counting cells in a larger volume of fluid than what we are interested in, providing an artificially high count. Given this, for each of the little grids, you do count the cells touching the left and top edges of the grid, and you don't count those on the bottom and the right. Based on these rules, we would have a total count of 26 cells in this small grid. We would then go through and count the other 4 small grids and add the total number into the table in your lab manual. Now let's try this with our sample. Place the hemocytometer under the microscope and bring it into focus at low power. You will see a variety of grids. For the red blood cell counts, we are interested in the smallest grids in the center. Here are the 5 grids we will count. Let's start with the bottom right grid. Zooming in to a higher power, we can take a closer look at the individual cells. Following the counting rules established earlier, I have placed a red dot on each cell. If you're doing this in a lab, you will use a hand tally counter to keep track. There is a total of 86 cells in this grid. Now I will use the same technique to count the other 4 squares. So here are my counts, and now I've added them up as well, and the total comes to 474. This number can now be placed in the table in your lab manual and then used for further calculations. Now we've reached the end of part 2, and we will move on to part 3. Thank you.